Good morning. Thank you for giving me this opportunity to present our work. Okay, so as most people here are aware, loss of function mutations in the gene which encodes for Tafazzin results in the development of Barth syndrome. Some of the most commonly reported clinical features of Barth syndrome include cardiomyopathy, skeletal myopathy, and cyclic neutropenia, as well as growth deficiency. So most patients with Barth syndrome have growth deficiency, so for both height and weight. Recently, growth curves have been developed. It was established that the 50th percentile for height of Barth syndrome patients, shown in red, is shifted down below the 10th percentile for the general US population, indicating that the average height for Barth syndrome patients is significantly reduced. So similarly for weight, the 50th percentile for Barth syndrome patients, again shown in red, was shifted down below the 10th percentile for the general US population, indicating that the average weight for Barth syndrome patients was also significantly reduced. So in fact, nearly 60% of individuals with Barth syndrome have a weight which is below the 5th percentile. There's a corresponding reduction in body mass index indicating that the average weight loss or reduction is irrespective of height. There is also a history of failure to thrive in 54% of patients. So together, these findings indicate that some individuals with Barth syndrome might display what might be called a skinny phenotype. So body weight is controlled by both food intake and energy expenditure, and as reflected in the amount of fat mass and the amount of lean body mass. So lean body mass is comprised of skeletal muscle, visceral organs, and the bone. So also because of the relationship between weight gain, lipid accumulation, particularly in non-adipose tissues, and a lipotoxic effect on insulin signaling, we looked at the development of insulin resistance in this study as well. So the purpose of this study was to determine how Tafazzin deficiency reduces weight gain in mice using the doxycycline-inducible knockdown. So the experimental model consisted of male mice, which were transgenic for the short hairpin RNA directed against Tafazzin using doxycycline to induce the knockdown, and their non-transgenic wild-type litter mates. So we used the 625 mg per kilogram diet to knock down Tafazzin in this model. So these mice were divided into 3 groups. The first group was fed a doxycycline-free diet. So in this group, the transgenic mice still expressed Tafazzin. The other 2 groups were fed doxycycline for either 4 or 10 months to induce the knockdown for the respective amount of time. So the first thing that we want to know was, does TAS deficiency promote this skinny phenotype? So we determined that the transgenic plus dox TAS knockdown mice were significantly smaller than that of their littermate controls. The growth curve for the transgenic plus dox TAS knockdown mice, shown in yellow, was shifted down below the dox-free groups, shown in black and red, as well as below the non-transgenic plus dox TAS wild-type mice, shown in green. So the TAS knockdown mice were significantly smaller than the dox-free groups, starting at 4 months of age, and this increased to a maximum of 25% smaller at 10 months. And the difference was more dramatic when we compared them to the littermate controls. They were significantly smaller starting at 2 months, and it increased to a maximum of 40% smaller at 10 months of age. So how has the body composition been altered to achieve this skinny phenotype? So first we looked at bone development. We determined that genotype had no effect on tibia length, and there was no difference in bone mass density between the various groups as determined by DEXA scanning. We also used DEXA scanning to determine the amount of fat and lean body mass in the transgenic and non-transgenic mice with and without doxycycline. At 4 months, we determined that the non-transgenic plus dox wild-type mice, shown in green, had increased weight gain due to increases in both the amount of fat and lean body mass compared to the dox-free groups. TAS deficiency reduced this weight gain, primarily due to reduction in fat mass, as there was no difference in the amount of lean body mass. At 10 months of age, the non-transgenic plus dox TAS wild-type mice, shown in green, had a dramatic increase in fat mass compared to the dox-free groups, and TAS deficiency again reduced the weight gain primarily due to reduction in fat mass. There was only a small but significant reduction in lean body mass in these mice. So to confirm the reduction in fat mass in the TAS knockdown mice, we measured the size of various fat pads. We determined that at both 4 and 10 months of age, the transgenic plus dox TAS knockdown mice had 50 to 70% reductions in the gonadal, perirenal, and brown adipose tissue compared to their littermate controls. So the TAS knockdown mice were smaller due to a reduction in fat mass. So then we wanted to see if there was a reduction in non-adipose tissues as well as a reduction in the adipose tissue. So we looked at triglyceride levels in the skeletal muscle, and we saw significant reductions in TG levels at both 4 and 10 months of age compared to their littermate controls. And the level was comparable to that of the dox-free group. Similarly, in the liver, we saw a reduction in triglyceride levels in the TAS knockdown mice, this time only at 10 months, compared to the littermate controls. Okay, so we're seeing a reduction in lipid in the adipose tissue, also in the non-adipose tissue. And because of that relationship between the lipid accumulation in non-adipose tissues and the development of insulin resistance, we did look at insulin sensitivity. So we performed an insulin tolerance test in these mice. So we fasted the mice for 4 hours, injected them IP with insulin, measured plasma glucose levels over time. So the transgenic plus dox TAS knockdown mice at both age groups, 4 and 10 months, had plasma glucose levels which were similar to the dox-free groups, so all those groups are down here, while the 10-month non-transgenic plus dox TAS wild-type mice shown in the black squares had significantly higher plasma glucose levels at each time point, showing, and in fact there was no reduction following the injection of insulin, indicating the development of insulin resistance. So the TAS knockdown mice were able to maintain normal insulin sensitivity. So how does TAS deficiency prevent this weight gain? So first we looked at food consumption, and we determined the food intake per gram of body weight in a 24-hour period. And we determined that the transgenic plus dox TAS knockdown mice at both 4 and 10 months were consuming significantly more food compared to the littermate controls. So the mice weren't smaller simply because they were eating less food. Okay, then we looked at energy consumption using whole body indirect calorimetry. We determined that oxygen consumption in the transgenic plus dox TAS knockdown mice was similar to that of the dox-free groups, but significantly higher than that of their non-transgenic plus dox littermate controls. We found a similar pattern when we looked at heat production. Heat production in the transgenic plus dox TAS knockdown mice was higher both during the day and the night compared to the littermate controls, but similar to that of the dox-free group. So the TAS knockdown mice had elevated energy expenditure and an increased metabolic rate. We also calculated activity levels and saw that the transgenic plus dox TAS knockdown mice had elevated activity compared to their littermate controls. Okay, so we've now established that hypermetabolism promotes the skinny phenotype in the TAS knockdown mice, and this was characterized by a reduced size of the fat pads. There was less lipid accumulation in the non-adipose tissue, and they had normal insulin sensitivity. So then we investigated the mechanism by which these mice had reduced lipid accumulation. So first we looked at white adipose tissue lipolysis. We determined that the fasting plasma non-esterified fatty acid levels were similar between the dox-free group to that of the transgenic plus dox TAS knockdown mice, but the level in the knockdown mice was higher than that of the littermate controls. The increased lipolysis was due in part to the adipocyte triglyceride lipase, or ATGL. So the protein level was higher in the transgenic plus dox TAS knockdown mice compared to the littermate controls, but similar to that of the dox-free group. So if there's increased fatty acids if there's increased lipolysis and increased fatty acids being released from the adipose tissue, where are they being used? So to answer this question, we looked at isolated skeletal myocytes their oxygen consumption using fatty acid as a major substrate. And we determined that both basal respiration as well as respiration due to leak or heat was reduced in the knockdown mice compared to the littermate controls. So skeletal muscle that was deficient in tafazzin did not have an increase in fatty acid oxidation. We performed the same experiment using isolated hepatocytes, and in this tissue we saw an elevation in both basal respiration as well as respiration due to leak compared to the littermate controls. So the TAZ-deficient liver had elevated fatty acid oxidation. And this was consistent with our whole body indirect calorimetry, where we saw elevated oxygen consumption and heat production. So do the changes in fatty acid oxidation correlate with the L4 cardiolipin levels? So we measured L4 cardiolipin in both the liver and skeletal muscle. So in these tissues, we did see a significant reduction compared to the littermate controls. However, in the liver, the L4 cardiolipin level was still similar to that of the normal DOX-free groups, but in the skeletal muscle, the level was reduced well below normal. So there was this correlation between normal hepatic L4 cardiolipin levels and the elevated fatty acid oxidation. So how does the TAZ-deficient liver promote both of these things, fatty acid oxidation and L4 cardiolipin synthesis? So we looked at mitochondrial trifunctional protein alpha, which catalyzes both fatty acid oxidation and cardiolipin reacylation. And we determined that the protein level was higher in the knockdown mice compared to both the littermate controls as well as the DOX-free group. So interestingly, we determined that there was no change in skeletal muscle MTP alpha levels. Okay, so our working model is that in the non-transgenic plus DOX wild-type mice, there's excess fatty acids which are being taken up by both the adipose tissue as well as the non-adipose tissue. And this accumulation of triglyceride is having a lipotoxic effect on the insulin signaling pathway. While in our TAZ knockdown mice, and the transgenic plus DOX mice, the TAZ-deficient liver is promoting the hepatic expression of MTP alpha, which promotes fatty acid oxidation and cardiolipin synthesis, which further promotes oxidative phosphorylation, ATP generation, and heat production. So the excess fatty acid is being directed away from triglyceride accumulation and resulting in this skinny phenotype with normal insulin sensitivity. So to summarize, the TAZ knockdown mice display a skinny phenotype. So it's characterized by reduced mass, TG levels in the adipose muscle, liver, and normal insulin sensitivity. And this is due to elevated oxygen consumption, heat production, and activity. We have elevated white adipose tissue lipolysis and fatty acid oxidation in the liver. So we've concluded that hypermetabolism and altered triglyceride metabolism is contributing to the low weight in the TAZ knockdown mice. So we have many collaborators. So I must thank my supervisor, Dr. Grant Hatch, as well as many other collaborators who have contributed to this study, including Genevieve Spragna, who did the mass spec, very talented former graduate student, Edgar Mejia, and excellent technical work by various other members who did DEXA scanning, genotyping, and performed some other experiments, which we didn't show here today. So thank you.
Great, thank you, Laura. So we have plenty of time for questions.
Hey, Laura, great talk. Of course, I'm very interested in this, right? So the tissue specificity of this effect, so skeletal muscle versus liver is really interesting,
Right.
and the TFP as well. So I'm curious, though, if you looked at those were mitochondrial respiration experiments, the picomole per mg, where you showed fatty acid oxidation?
Those were whole cell.
Oh, whole cell, okay.
Yeah.
Is there a way to get at coupling of oxygen consumption to ATP production? And so my question is whether, if you think there's more uncoupled fatty acid oxidation in the liver just to get rid of this excess fuel, and why you think that would be in the liver but not at all. It seems to be the opposite, right? I mean, reduced capacity to oxidize lipid in the muscle.
So by uncoupling, you mean like the addition of FCCP to achieve maximal, or
Well,
just
whether or
peak
not,
in general? So we did measure heat in general, which is what we show leak, oxygen consumption, which is associated with leak. So we did see a reduction there. Maximal wasn't altered for the liver. It was reduced in the skeletal muscle.
Okay, so that would be sort of a whole cell assay of uncoupled respirations of heat. Okay, interesting. Okay.
Whole body.
We did whole body heat, yes.
So it's uncoupled whole body.
I wanted to confirm because of course heat production is also associated with what we would call coupled. I mean, no one's perfectly coupled, so you're always going to see increased heat production with activity, right? But if it's more per O2 consumed versus control, and if that's what I'm understanding, then that would certainly be what you're saying. That's
Right,
cool.
yeah.
Yeah. Thanks.
So I have a question here. So very interesting work. The elevated body temperature, did you look at the brown fat?
Yes.
And also the thyroid hormone level in those mice?
Which hormone level?
Thyroid hormone. Thyroid
hormone.
hormone, yeah.
No, we didn't measure thyroid. We did measure adrenaline and noradrenaline, and they were higher in the TAS knockdown mice. Leptin was lower, adiponectin was higher.
The brown fat?
Brown fat, we didn't measure oxygen consumption from brown fat, but we did not see any change in uncoupling proteins in any tissue that we measured, actually.
The
In
level
protein
of brown
levels.
fat, level of brown fat didn't increase?
Level of brown fat was reduced.
Reduced?
Yeah.
Oh, thank you.
Yeah.
I have very simple question. In your experiment, how did you do your induction with ducks? That's show food or sucrose water?
How did we do the TAS knockdown?
Yeah.
We did just feeding and chow, the 625 mgs per kilogram. We induced, so we put the dams on doxycycline prior to breeding them. So the females were preloaded prior to being pregnant.
I mean, which way? That's sucrose water or?
No, there's no sucrose water. It's in the food.
Just in the food,
Yeah.
chewing food. Okay, thank you.
Great talk, Laura. But I'm still a bit confused about this increased respiration due to leak and what that has to do with fatty acid oxidation. First of all, did you show enhanced beta oxidation in the liver? And secondly, fatty acid oxidation itself has no correlation whatsoever with leak. So if it's a leak, then there has to be some UCP-induced leak or something. So how do you reconcile these two things?
Well, we see increased basal and increased oxygen consumption from the leak. So when we measure basal, there's two parts. There's ATP-dependent respiration, and then there's always a leak-associated respiration, right? So each time we measure basal, it's split up like that. So when we had an increase in basal, there was increase in ATP, and there was also a corresponding increase in leak. So all the ratios were maintained. All of it was increased all at the same time. So it just looks like there's more flux through the oxidative phosphorylation pathway.
But if you say leak, that's oligomycin inhibition?
Right. No, oligomycin inhibition is ATP dependent.
No, the oligomycin
So it's
blocks
the
ATP synthesis.
non-oligomycin
Oxygen consumption.
remaining, right, respiration.
Okay,
If that
but
makes
I'm still confused. Okay, we'll discuss
We'll
it
have to
later.
talk later, yeah.
Just a comment about the conditions. So first you have somewhat fat, and the dox actually makes the mice bigger.
Right, yeah, this is an antibiotic
Versus
effect.
lean.
Yeah.
So you're actually not testing the exact same environment. And the second comment is that if you maintain the mice at typical laboratory temperature, that's actually a stress, cold stress for the mice.
Okay.
Because their normal environmental temperature is much higher than our room temperature. So
Right.
some of these effects may not be task specific as much depletion specific as they are the environmental conditions and the difference in the size of the mice.
Okay, sure, yeah.
Yeah, we'll have to talk afterwards because we see in humans actually increased fat mass.
I'm aware of
and
that.
lower lean mass. So I'm wondering if it's different as they get older and they get less sedentary. And also when we do indirect calorimetry, we don't see any differences. In fact, when we see resting energy expenditure is usually lower, and we think it's just because they have such lower skeletal muscle mass. But we can talk about that afterwards. Okay. Great. Thank you so much.
Okay, thank you.